Thank you, Ran and David, for this opportunity to speak in this most excellent venue. So I'll be talking about place cells and place codes in hippocampus. And this work is done in the lab of Yoram Burak. I'm a PhD student there, and it's in collaboration with Rava. In the screen, you see a QR code, and you can find the link to the paper, which was just recently out a few months ago. So with that, I will start. So I will start with a brief outline of what is going to be in the talk. Firstly, we will do a brief review of background and experimental results in place fields, and this will focus particularly in irregularities observed in place fields measured in large environments. And I will be focusing on 3 particular papers that I will follow throughout the talk. The motivation here is also to raise a question about the diversity of features of these place observations in place cells and if they can be understood within a given model. In the second part, I will introduce our model, which is a model of Gaussian process thresholded that generates place codes. And I will introduce the model itself, briefly go over the mathematics behind it, and most importantly, introduce the main parameters of the model. And once we do that, we will move on to analyzing in detail various predictions of the model. First, we will look at predictions of the model for arrangements of the place fields. By arrangements, I mean things like the sizes of the place fields, the gaps between them, where they are in the space, and so on. And in the next section, I will go in a more detailed prediction about the shapes of the fields themselves, their geometry and topology. And once we do that, we will kind of reflect back on the model and try to think about mechanisms that might give rise to such a model, and also have some normative considerations around efficiency and what the function of such a neural code might be. So with that, let's start. Firstly, before going into irregularities, I want to talk briefly about place field regularities. And by here, I want to introduce what I call the classic view of place fields. And this is a viewpoint primarily motivated by experiments done in very small environments. And these were the environments where place fields were first identified. And here I take some sentences from these from a review paper from Bob Muller and John O'Keefe, and find that they say things such as place cells generally have rarely have more than 1 fields. They have single maxima. They are mostly circular or elliptical. And if there are any variations, that might just be some kind of noise. And indeed, experimental observations in small environment does give rise to a viewpoint like this. But recent, more new experiments kind of question this viewpoint. And here, there are many new experiments, but I'll focus on 3 particular ones. The first one is an experiment done in the lab of Nakhimulanovsky, where they measure bats flying in a long 200-meter long tunnel, and they measure the neural activity during the bat's flight. The second experiment is done in the lab of Jean-Marc Velous, where they measure hippocampal activity in rats, kind of exploring this large mega space. They call it 18.6 meter square arena. And here they have a nice setup where they have a robot that dispenses snacks so that the rat kind of uniformly covers the space. And finally, we'll also look at a little older work from Nakhimulanovsky lab again, where they measured bats firing fields in volumetric environments, so 3D space in an enclosed room. The results of these papers can kind of be summarized along dimensions. So there is measurements of bats in 1D environment, measurements of rats in 2D environments, and measurements of bats again in 3D environment. And we're going to look at these results in some detail. So firstly, kind of going back to the classical point of view, this is what you would see if you measure hippocampal place cells in CA1 in an environment of length 60 meter. That is a 60-meter portion of the tunnel. And here the colors simply mean 2 directions of the flight. There is remapping, so the place fields are different in 2 directions of the flight of the bat. and the colors simply mean the two directions. So here you see an example of a cell that has a very nice parabolic looking field in this small environment. Now, when we go to a larger environment for the same animal, that is, we go to something like 200-meter long environment, we start seeing place fields that look like this, or tuning functions that have multiple place fields. And each place field is different than the other in terms of its height, width, and also kind of its shape in general. These are raster plots on the bottom, and the firing rate curves, again, two directions. And here they want to make a point that even within a given cell, the ratio of the smallest to largest field is quite big. So there is really a lot of irregularity. Now, this irregularity kind of becomes regular when you measure all the place fields in a given cell and across the whole population of cells and draw a histogram. And there you see this nice skewed distribution, which in the paper, they kind of heuristically fit to a log normal or a gamma distribution. And at the same time, they can look at the gaps between the place fields, and they find this nice fit to exponential distribution. So this is kind of 1D. Now in 2D, we get a similar situation kind of qualitatively, where in small 2-meter square arena, you get these really nice classical place fields. But when you go to larger environment, you start getting situations like this, where there are multiple fields and shape of the place fields themselves are variable. And if you, again, look at the histogram of the area of the field sizes across all the cells, you get a distribution like this. It's a nice looking distribution, but I want to make a point that it is in contrast to the distribution in 1D, which looked completely different. And if you measure number of fields per cell, here too, you get in larger environment sizes, you get cells with really large number of fields, and on average, they have more than one fields. Finally, we have 3D measurements. There's only one in environment size, so we can't contrast small to big, but here we generally don't have recordings from 3D volumetric environments, so we can think of this as like an extension to classical kind of environment. And this is the kind of firing rate cloud you see of a firing field of a given place cell. And one can try to kind of segment them into different place fields and so on. And in this plot, they show if you project them, so you take a place field and you look the size of this place field along the x-axis, y-axis, and z-axis, and measure the histogram again, you get this also the kind of skewed distribution like we see in the 1D case. So this is, again, projected first along axis and then measured. And if you measure number of fields per cell, then too you have many place cells with multiple place fields. So this is a kind of whole picture of really diverse and irregular firing patterns and structure in the place fields of place cells. And it kind of raises a question if we can do better than classical uniform tiling viewpoint of place cells and combine these things within the purview of a single model. And now we're going to propose one such model. So I will describe the model in one dimension because it's the easiest to motivate and easier to see, but it holds for any dimension, so 1, 2, and 3. So what do we want? We want place cells to encode 1D space and a continuous variable, basically coordinates of animal in the space. And in the experiments that we're looking at, we want to encode a big space, let's say of size L, and the question is how. So in our model, we propose that a neuron's spatial code is generated randomly and kept fixed. What do I mean by that? I will make randomly more precise, but basically we imagine a neuron's spatial code over a given environment is generated randomly, and then it's frozen. So every time the animal enters that space, it will have the same spatial code. So here we need to make randomly more precise. It's random over a continuous coordinate. So a random function over spatial coordinates, a continuous coordinate, is a stochastic process. And I think I want to take a second here to kind of avoid a confusion. because stochastic process in theoretical neuroscience is generally used in the context of dynamics over time of neural networks, and that is not the context it's being used in here. I'm talking about a random function as a function of spatial coordinate, that is the location the animal is in, and it gives rise to the spatial code of the cell of a given place cell. So I'll make it more precise now. In particular, we model the subthreshold activity of a place cell as samples from a Gaussian process. And I will give a brief primer on what a Gaussian process is so that we're all on the same page. So before going into continuous space, let's start by discretizing the space. Think of this line on the bottom as x-axis of some space, and we discretize it. And for each discrete location, we sample some random value from a Gaussian distribution with the condition that nearby values be correlated. So the value here is correlated to this value here. And there is some distance up to which it's correlated, and then the correlation falls off. So if we do this discretization and sampling in a finer and finer way, at the limit, we get to a continuous function, random function over the space. And this is going to have a property that if we pick any finite number of points in the random function, it will be jointly Gaussian distributed. And this is exactly what a Gaussian process is. Now, similar to Gaussian distribution in like 1D or multivariate case, a Gaussian process, which is a random function, also needs two things for its definition: a mean function and a covariance function. For us, mean function is a constant and not important, and I'll explain in a bit why it's not important. But what's more important is a covariance function. And the question is which one? One can come up with various choices for a covariance function for a Gaussian process. Here we have this nice property for our model and the kind of things we're interested, that the covariance that we can choose is very general, as long as a covariance function obeys two mild property. First, that it decays, meaning that a point in some location is not correlated to a point very far away from it, and a covariance function that has a second derivative. So this is kind of a technical regularity condition, but it's simply to avoid the situation where our Gaussian process is something like a Brownian motion or Ornstein-Uhlenbeck process, where we cannot define a derivative. So as long as these two mild conditions are satisfied, all the results that I'm going to show to you hold. Still, we do need some particular moments or properties of this Gaussian process to understand them properly, at least in the context of our model. And the first is simply the variance R. So square root R kind of defines the size of this Gaussian fluctuation, and R double prime is the derivative of the, it's the variance of the derivative. And why is it important? It's kind of a weird quantity if you think a priori, but if you think about it, the ratio of variance and variance of the derivative sets a spatial scale. So here, as the variance of the derivative gets large, this quantity, the correlation length as we call it, gets larger, and we start getting smoother Gaussian process. When it's smaller, when the variance is larger and the correlation length is smaller, we get processes like in the top where it fluctuates a lot. So this quantity helps us set a spatial scale, and it's in the unit of meters. So these are the two parameters of the Gaussian process itself. And to complete the model, we finally introduce a thresholding parameter. So it's going to be theta in the length scale of the standard deviation. And we threshold the process at this value and simply linearly scale it. So it's a ReLU over the Gaussian process. And it gives rise to the firing fields in the model. And depending on the correlation length and the thresholding, here are three different examples of how the firing fields of a neuron can look like. In the top, we have a case where there are multiple fields and they're more heterogeneous. And in the bottom, we have a case where there's more classical looking parabolic fields and only a few of them. Once we do this setup, we can now start studying the model kind of analytically and mathematically. And there are various tools for it. So firstly, these components, the firing fields, individual firing fields themselves in the literature of Gaussian process are called excursion sets. So basically, these are parts of the process that go on excursion above the threshold and come back. And various properties of these excursion sets can be studied exactly. So on the left is distribution of count. So what is the distribution of number of such excursion sets or number of fields? This can be calculated exactly as a Poisson distribution with the rate given by Cacciari's formula. So Cacciari's is a theorem that allows us to study threshold crossings of stochastic process in a very exact way. Now, if we want to study the distribution of sizes, which is what we're more interested in, it gets a little complicated. There is no general expression for it, but assuming large threshold, so assuming threshold becomes very large, we can approximate the distribution of field size as this expression on the right. So there are two things to note on this expression. First, it has one parameter beta, which is simply set by the mean of the field size. And if you think about it, just number of field counts along with the time the field spends or the portion of the space field spends above the threshold can give rise to the average size of the field analytically. I will not write the expression here, but we can get this single parameter which kind of depends on the correlation length and threshold in a degenerate way. And more interestingly, you'll note that this expression has d here, which is the dimension of the space, meaning that the distribution of field sizes has a dependence on the dimension that the animal is in. So in d equal to 1, that is in the tunnel, for example, it would give a different distribution as opposed to d equal to 2 that is in an arena. So with that, we complete the setup of the model, and now I will go into looking at the detailed prediction of the model and also kind of matching it with the data and validating the predictions against data. So here is the general kind of procedure we'll follow. This is an example of just a real place field of a bat on the top and a sample of how place fields look in the model. And the procedure to kind of quantitatively study it is first we start with the parameters. There are two parameters of the model, correlation length and threshold. And we will fit this to each experimental data set, that is measurements in a given environment size. And once we fit that, we get a distribution over various features of the model. And this is because the model we have is a generative model. It generates samples for how the place field should look like given sigma and theta. So we can measure any observable of place fields generated in this way and understand the probability distribution of that measure. And for example, the field size and gaps between the fields. So to make it more concrete, we'll start, for example, with the data of bats flying in 200-meter long tunnel. So just to set the convention now for the rest of the talk, data is always in green. The model will be in orange, and the overlap of data and model will be brown. So here I'm showing two histograms measured in place fields of bats flying in a 200-meter long tunnel. This data was given to us by Nachum Lanovsky lab. Once we have the distribution, we will simply infer sigma and theta by matching the means. Not the whole distribution, but simply we'll calculate the mean field size and mean gap size. And that is sufficient to infer sigma and theta either through simulation or even analytically. Now, once we infer it, then the model generates a prediction for what the field size distribution should be like. And that's what you see in orange here. You will note there's a slight discrepancy between the simulation and the analytical theory, and that's because the theory is only exact in extremely high threshold. We're already in high threshold limit, but there's still a gap. But the important quantity here is the histogram arising due to simulation. At the same time, the same parameter will also give rise to a fit to the gap distribution, the gap between the place fields. I think the right distribution, the distribution of field gap, is less interesting. It's basically saying the location of place fields are independent. And that makes sense. If the correlation length is not too large and threshold is large enough, the location of these threshold crossings in the model will be independent. So they will simply be distributed as exponential distribution. What's kind of more interesting and non-trivial is the distribution on the left, which is roughly a Rayleigh distribution. And this distribution has this kind of characteristic skewed structure. And in particular, it has this dislike for really small field size. And this is a general property of smooth Gaussian processes. It's called the repulsion between threshold crossings. So basically, if you look at a field and the two edges of the field, they tend to, on average, statistically repel each other. So they really don't like to be close to each other. And that is why when you go close to very small field size, you get a sharp decline in the counts of fields with small field sizes. At the same time, field sizes also cannot be too large because it's statistically unlikely that the field will spend very long portion above the threshold. So you also get a sharp decrease in the probability of having very large fields. And together, these two facts give rise to this kind of characteristic shape of the field size distribution. Now, as I mentioned earlier, previously, this distribution has been kind of heuristically fit to log normal distribution. And we're kind of in a position where we can really test or compare this with log normal in a very quantitative way. So here's how we do it. If the distribution was log normal, then log of the field size is a normal distribution. This is what being log normal means. And normal distribution has the property that its higher moments are 0. For example, skew. So what we do is we take log of field sizes in the data and calculate its skew. At the same time, we also do it for histograms generated by multiple simulations. So we simulate until we generate the number of fields matching the data. We get one histogram, we measure the skew, and we repeat the procedure. So we get a distribution of skew, both for the log normal model for place fields and the thresholded Gaussian process. And here what you see is these histogram and the skew measured in the data, which mostly is very consistent with the skew in the model. And we can also do that for even higher moment, kurtosis, and see that the match is much better with the thresholded Gaussian process model. So this is just to establish that we not only capture kind of what you see here, means and a kind of rough shape, but also in a detailed kind of quantitative higher moments of the distribution. Now that was 1D. We can repeat the procedure for 2D. In here, on the left, you see an example of a real place field measured in rats. And on the right is a sample from a model where we simulate 2D Gaussian process and threshold it at some value. And kind of you can already appreciate qualitatively there is some similarity, but we can similarly look at more quantitative predictions and find that the distribution of field sizes, in fact, matches the distribution of field size observed in data. And it's exponential because if you substitute d equal to 2 in the equation that I showed you earlier, you end up getting the distribution of field size in 2 dimension is exponential. And field counts are simply distributed Poisson. Finally, we can repeat the same procedure for 3D data. Here on the left, again, we have a sample of a real measured place field in bats. And on the right is an example of how place fields look like in simulation of the model. And in 3D in particular, we are in a position to do a very detailed kind of study, which is if you take a 3D thresholded Gaussian process and you take slices, you take 2D slices of this 3D fields, those themselves follow 2D slices follow 2-dimensional thresholded Gaussian process statistics, and 1D slices follow 1-dimensional thresholded Gaussian process statistics. So firstly, we can measure the volume of these quantities and substitute d equal to 3 in the expression that we had before, and we get a nice fit with the distribution of receptive field volumes. Then we can look at 2D slices of the receptive fields, both in data and model, and measure the receptive field area and substitute d equal to 2, and we get the exponential distribution. Finally, we can repeat the procedure with d equal to 1 and get Rayleigh distribution. And this is a nice way to validate the prediction for the field size distribution across the values of d going from 1 to 3. Cool. So far I've been focusing on like one experimental data set in 200-meter long environment, but a natural question arises: what happens across environment sizes? So next we'll look at parameter scaling as a function of environment size. The data was most abundant in the environments that I looked at, but also in other environment sizes, we can infer these two parameters, sigma and theta. And what we find is this roughly power law-like trend in the increase of correlation length with the size of the environment. So it's not a many points to actually say it's a power law, but something we can clearly see is that it's sublinear. And the effect of the trends in correlation length, along with the trends in threshold, which is much higher in smaller environment and smaller in larger environment, is that field counts, that is number of field per cell on average, grows sublinearly. So basically what's happening is as the environment gets larger, correlation length also gets larger, but it gets larger sublinearly, and threshold goes down. And the effect of these thing is field counts go up, but propensity, which is fields per volume, so number of fields observed per unit length, goes down, but weakly. So it changes very slowly, but it does go down with the size of the environment. So that was all about how fields are arranged and the statistics. The next section is going to be about field shapes. In this section, we're going to study kind of some detailed properties of the geometry and shapes of the field. And the first one is kind of the most intuitive and easy one to ask. Basically, in large environments, we see fields have multiple maxima. How common is that? Here, we simply use the same parameters that we inferred by matching the field arrangements and calculate the distribution of fields with 1 maxima, 2 maxima, and 3 maxima, and measure it against and compare it against with data. And we see it roughly captures the frequency. And I think a point that I should kind of stress here is for all the results, including this one and results from here on, we don't do any further parameter fitting. So in some sense, the moment we fixed sigma and theta to match the statistics of arrangements of the field, the structure of field shape is completely determined. There is nothing further to do to understand the structure of place fields and their geometric property. It's simply already set by the parameter that fixes the arrangement in space. The second field shape related property that I personally really like is distribution of field slopes. So at the boundary, we can measure the slopes of the fields. And for Gaussian processes, it's known exactly, not in any approximation, exactly that this distribution follows a Rayleigh distribution. And here I'm taking kind of absolute value of this, and you see the match between data and model is really excellent. And also analytically, because this is not an approximation. Next, there are these anecdotal observations that larger fields tend to have larger amplitude. So we wanted to understand the joint distribution of field size and amplitude, and this is what's shown here. So on average, the relation between the field size and amplitude follows a power law. And it also captures the spread of the joint distribution pretty well. This is in 1D. And we can repeat the same procedure in 3D. And we also get a power law distribution with a different coefficient. And finally, in our kind of study of field shape, this is maybe the strongest evidence for the Gaussianity or the thresholded Gaussian process model, which is the Euler characteristic. So I will start by looking at Euler characteristic in 1D, which is a simple case. In 1D, simply Euler characteristic counts the number of connected component. And in our case, that's simply the number of fields. So here's what we do. We start with the fields and we start to progressively threshold them. So we take the fields in the data and we add more threshold and count again, add more threshold and count again. So the number of fields go down in a characteristic manner. And this is known exactly, and we have an equation for it, which simply requires the sigma and theta, which we have already inferred before. And that is this orange curve. And if we do this measurement in the data, this is how it looks like. Now, we can repeat this also for 3D case. But in 3D case, we need to be more careful because we account for not only connected components, but holes and voids. So when you go to 3D place fields, there is possibility that the fields have a hole in them, and the holes themselves can be 1D or 2D. So together, this component gives a kind of non-monotonic characteristic to Euler characteristic. And this is what it looks like, analytic prediction, and this is how the data looks like. Now, what exactly are we capturing here? We can try to look at an example of fields with high and low threshold. And these are some examples I've taken from a paper published in astronomy. And there is an interesting link here, which is that various experimental and theoretical work in measurements of cosmic microwave background radiation have used exactly Euler characteristic to verify or refute Gaussianity of certain measurements that they do at the cosmological level. So, and the reason behind this is the Euler characteristic for different stochastic processes are very different. So you really get a fine-grained sensitive distinction between different stochastic processes. So if you want to say this is a Gaussian process as opposed to some other process, Euler characteristic is the way to go. Great. Now, again, going back to the question of scaling, we looked at how things differ in scaling in arrangements of field, but can we say something about scaling of field shapes across environment size? And the main difference that matters here is threshold. So for example, in the bats data, in small environment, you end up seeing very high threshold, 2.4. And in large environments, you get low threshold, 1.1. So here is the effect of these things. In the model, I sampled randomly 16 place fields with threshold 1.1 and 2.4. And what ends up happening is basically at the limit of high threshold, you get this strong tip of iceberg effect and all the fields look parabolic. And this is not just a qualitative statement. You can quantitatively say that fields in high threshold limit must look parabolic. While in a slightly lower threshold, like for example, 1.1 in 200 meter, you get fields that have more exotic structure with multiple peaks and leaning towards one direction or another. And if we think about it, this explains the observation that classically place fields were imagined as uniform parabolic field that tiled the space. And that's simply because in small environment, threshold is high. And indeed, the place fields that you saw were parabolic. And now when we can do measurements in much larger environment, the threshold goes down and we start seeing these more exotic curves that reveal the underlying heterogeneity. And that brings us to the close of all the predictions. And now we will go to the reflection phase of the model. Wait, before that, maybe I should also talk about parameter heterogeneity given time. Okay. So throughout this model, I've been talking about sigma and theta as fixed quantities, same for all the neurons in the population, but it need not be like that. What if there is some heterogeneity? With correlation length, it's hard to see the impact of the heterogeneity, but with threshold, it's very clear. And here I'm trying to show a schematic. Imagine a bat flying in one direction versus another, and there is a cell with threshold theta 1 and another cell with threshold theta 2. And what will end up happening is the cell with higher threshold theta 1 will end up having just one place field in both the environment, whereas cell with theta 2 will end up having two place field in both the environment. So it induces a correlation between statistics of place fields, even though the underlying structure might be independent. And this is indeed what we see in the data. So in our simulation, we basically fit a skewed normal distribution. It doesn't matter exactly what distribution a threshold will look like, but what we can see is that it gives rise to a correlation between the field count in forward and backward direction in the bats. And this is indeed observed in data, that there is a correlation and we fit the correlation value. And we can also do that similar thing for the field sizes in forward and backward direction. And there is also a correlation arising due to this diversity in thresholds. Now, one nice side effect that we didn't anticipate out of that is that the fit to the field size and field gap, in fact, get better. So if you remember earlier, the field gap distribution had somewhat of a bad fit at the very tail, and that gets much better once we introduce variability in the thresholds of cells in the population. Okay, so with that, I will just now go to the reflection phase and try to think what kind of mechanism might lead to a model like this. So in our thinking, the model is we have spatially selective place cells in CA32, and these are uniform, more classic looking fields, even in larger environments, as has been shown by Ulanovsky lab recently in their SFN poster, that even in large environments, CA3 place cells remain unimodal and tile the space. Now, if we take a place code like this in CA3 and project it randomly in a feed-forward manner to CA1, the input to CA1 will indeed be a Gaussian process, and the connections need not be Gaussian themselves, but we can invoke some sort of central limit theorem kind of result for random functions and see that the input will roughly be a Gaussian process in CA1. So there's two ways to look at it, one as a proposal for mechanism, and the other is to interpret the success of the result as there being a random connectivity from CA3 to CA1, or predominantly random connectivity from CA3 to CA1. The next angle on the model can be more normative and efficiency oriented. So we can ask the question, is it a good idea to have a model of a variable that's based out a Gaussian process? And for this, I'm gonna use the framework and develop on the results from this paper, 2022 paper from Yoram and Rava. And the setup is basically the following. We have a decoding framework. Imagine the animal is in some space and the coordinate is x, and it's being represented by a Gaussian process code. We will forget about threshold for now and add it later. So simply the spatial code, the neuronal spatial code is a Gaussian process, and it's being further corrupted by a noise during measurement time. And some downstream brain area needs to decode the location of the animal's position in the space using something like a maximum likelihood decoding. In this kind of framework, what we find is this result that the expected mean square discrepancy between the animal's location and inferred location using a Gaussian process code has two elements. The first part simply is the Fisher information or inverse Fisher information, and the second part is something that comes from what we call global error. And let me briefly motivate global error. So imagine there are 3 neurons in our population and they're trying to code for space. And we can have different correlation length, small correlation length to large correlation length. And immediately in the case of small correlation length, you see that the Gaussian process code can by chance have long distance catastrophic intersection. So there is a chance that the neural code will accidentally end up looking like the neural code of some location very far away due to the underlying randomness. And this can give rise to catastrophic global error. But if we make the code very smooth, we get this kind of smooth non-intersecting code at the price of neural code changing very slowly. So our resolution of the variable will be small. And I can make that more precise with kind of this schematic. So if we think about the first term here, which is the Fisher information, you can think about the posterior. So some brain area is observing from CA1 Gaussian process code, and it has a posterior around the true location x star. And the width of this posterior depends on the correlation length. When the correlation length is large, the width of the posterior is large, and we have kind of a bad resolution of exactly where the animal is in. So we can try to remedy that by making the correlation length smaller, but there is a price to pay. There will be now more peaks in the posterior, and in fact, there might be a peak which is in completely unrelated location in the environment, and we will end up inferring that animal is in completely wrong location. But when we do the calculation, we find that the probability of such global error is suppressed exponentially in Gaussian process-based code. This is a particular property of codes like a random Gaussian process or grid cell-based code, and classical kind of uniform tiling codes don't have this kind of property. And here I'm showing you two curves because we've been working on this model more and have a kind of improved expression for exactly how the global error should go. So there was a previous analytic expression using some approximation, and now we can do it more exactly and calculate analytically what the rate of global error will be in Gaussian process-based code. And this was for unthresholded Gaussian process. And we now have even generalized the model to a thresholded Gaussian process. And still, we have the situation where the global error rate is exponentially suppressed. So one functional viewpoint at this point could be that these Gaussian process-based code are optimized for efficiency. So kind of taking stock here, and I'm almost at the end of the talk. The mechanism, our reflections and mechanism and efficiency first kind of lead us to think that the connection from CA3 to CA1 though might be predominantly random. And more normative theories suggest that CA1 is creating some kind of efficient compressed representation of space that can be read out with high accuracy. And this gives rise to a few questions. Firstly, earlier we saw a certain kind of scaling behavior in the parameter, like correlation length. And it gives rise to a question, is this scaling arising due to some normative consideration where the code might be trying to balance local and global errors, and we're working in this direction? And it gives rise to a larger question, why is there this dual CA3 sparse and CA1 dense representation of space? Why is this dual structure of spatial representation in hippocampus? So with that, I think I come to the end of my talk, and I had three major acknowledgements. First is to Unalovsky lab, who gave us the BATS data and many conversations, and now we have ongoing collaboration with the lab. Second is Jean-Marc Fellows' lab, and they provided us with the rats data that we analyzed in the paper. And finally, to the community and the guidance in the Burat lab and ELSC widely. Thank you, and you can find the paper using the QR code.